immune checkpoint blockade, as we have also seen through this meeting, and as we learn over the last 5 years, can be very effective in patients with lung cancer. But we still see responses in 50% of patients who are treatment naive. And among patients who do respond to these therapies, we do see the development of acquired resistance over time. And so these are two of the challenges that we are facing when we think about immunotherapy for patients with metastatic non-small cell lung cancer. And so we really need to identify new biomarkers or optimize existing biomarkers for immunotherapy outcomes so that we can really inform rationally both trial design, but also inform clinical practice and how to optimize the use of current immunotherapies. Right now, we have learned that there are several factors that can mediate the development of acquired resistance to immunotherapies, and our group has presented some of these results at ASCO last year. But we do know that in patients who respond initially to immunotherapies and then eventually develop acquired resistance to these treatments, there may be some changes in the tumor that can be responsible for the development of resistance. We have specifically identified that in a cohort of patients of 82 non-small cell lung cancer, there were acquired mutations in genes previously known to provide for primary resistance to immunotherapy, such as STK11 or SMARCA4 or KEAP1. While we knew that these genes could be present at baseline and potentially associated with a low likelihood of response to immunotherapy, we learned in this study that this mutation can also be acquired at the time of resistance. We also noted that beta-2 microglobulin loss of function mutation were common at the time of resistance. In this cohort of patients, 5 and beta-2 microglobulin loss. And we also noted that the expression of HLA class 1 decreased at the time of resistance, suggesting that this could be another mechanism of acquired resistance to these immunotherapies. We also performed some other immunophenotypic analysis, try to understand whether the immunophenotype of these tumors also changed at the time of resistance. And what we found was that at the time of acquired resistance, there was a decrease in the density of CD8 positive T cells, but also the PD-1 to PD-1 engagement, which is the proximity between PD-1 and PD-1 positive cells, increased at the time of resistance, suggesting a limited engagement between PD-L1 and PD-1 at the time of resistance. So these are some of the differences that we found between baseline and resistant samples in patients with matched pre- and post-immunotherapy tumor biopsies. And this is relevant because has new therapeutics have been developed to target STK11 or SMARCA4, for example. This suggests that as we do in other subset of patients treated with targeted therapies, also in patients who receive immunotherapy, may be worth performing repeat tumor biopsies to determine what's the resistance mechanism and what we can do about it, whether or not we can act on it by enrolling patients in clinical trials, for instance. So I think these are some of the implications for this study.